My name is Daniela Mikovic. I'm coming from Belgrade, Serbia, and it is my pleasant duty to welcome you at this last session of the first European Congress on Thrombosis and Hemostasis. Today we are going to listen a plenary lecture about emerging gene therapy strategies in hemophilia. And please let me announce our speaker. Professor Thierry van der Driessche, who is full professor at Faculty of Medicine, University of Leuven and Free University of Brussels, where he is now director of Department of Gene Therapy and Regenerative Medicine. He served as the president of the European Society of Gene and Cell Therapy, and he is recipient of the Outstanding Achievement Award from the European Society and the Excellence in Research Award from the American Society of Gene and Cell Therapy. So, Professor van der Driessche, I kindly ask you to present your lecture.
First of all, I wanted to express my gratitude to be invited here. I'm really greatly honored to be the plenary speaker this afternoon, so thank you very much. This has been a great meeting, a lot of interactions. It's extremely dynamic. I pretty much enjoyed the session, the fast and furious session this morning. It's definitely the right spirit for this meeting, I would say. So what I would like to do today is to give you an overview of where the field of gene therapy for hemophilia currently stands, what are the challenges, and where we're going from here. Is gene therapy a magic bullet? Is it going to cure hemophilia? Is it going to be the ultimate treatment without any side effects? Well, that's what we hope, but I think we have to be cautious, and that would be the take-home message of my presentation today. In the year 2000, a historical milestone was achieved with the determination of the complete human genome sequence. It wasn't just important for medicine, it was important for humanity. It was truly a historic achievement. And in 2000, the then President Bill Clinton invited Craig Venter and Francis Collins as representatives for this worldwide effort to the lawn of the White House to celebrate this important event. And many of you are active in the field of GWAS and this type of analysis, so you understand better than I do the true implications of this very work. And I quote Clinton, who said that with this profound new knowledge, humankind is on the verge of gaining immense new power to heal. It will revolutionize the diagnosis, prevention and treatment of most, if not all, human diseases. Perhaps a little bit too ambitious, but nonetheless, the tone was set. And in a little bit more than a month from now, we may perhaps have another Clinton in the White House. Well, I hope it's not the alternative, who may have another vision or no vision at all. What inspired me to go into gene therapy is the fact that when a child is born with one point mutation in his or her genome, it has to bear the burden of that mutation for the rest of his or her life. So it's like a sword of Damocles that's hanging on a patient's head, and sooner or later, the patient will succumb from that very mutation. So just one point mutation, to put it in perspective, in 3 billion base pairs can cause a severe, potentially life-threatening disorder like hemophilia. Luckily, we have treatment now, but there are other diseases, monogenetic diseases, for which there is no treatment, like Duchenne muscular dystrophy, cystic fibrosis, and so forth. To put this into context, it's equivalent to one typing error, not just in one complete set of Encyclopedia Britannica, but 15 of these volumes. And believe me, I've done the calculation and it matches. So it's one typo, if you may, in 15 entire volumes of the Encyclopedia Britannica, and if it's at the wrong place, you basically have to suffer for the rest of your life. So can we fix it? Can we fix these genes? Well, there's two different approaches that we can employ to this effect. One could employ gene addition, which means the introduction of a functional copy, a normal copy of the gene, into a patient's cells that compensates for or complements the defective gene. So in that case, you don't really care about the defective mutated gene, you just add a functional copy. In a way, it's like driving with a flat tire and you just add a new tire to your car while the flat tire is still running. So this is definitely useful for recessive genetic diseases like hemophilia. There's another way of trying to fix a broken gene, and that's by gene editing. And that's correcting the mutation directly in situ within the defective gene itself. So it's like a form of molecular Tipex, if you may, where you edit the very nucleotide that is causing the disease. And this type of gene editing can be useful not just for recessive, but also for dominant genetic disorders. Just to emphasize it a little bit more strongly in a conceptual format, so if you have a mutated or broken gene, conventional gene therapy, and I can't believe I'm actually using this term because there's nothing conventional about gene therapy, but one can just add a gene and this functional gene will compensate for the defects of the broken gene. In the case of gene editing, you really try to fix the broken gene itself and replace it with a functional corrected copy. So in the course of my presentation, I'll talk about let's say the yin and yang of these two different approaches. Now, gene therapy started effectively in the early 90s, and the technology that was available in those days was not fully developed. And it all boiled down to the mantra, to paraphrase the mantra in real estate, delivery, delivery, delivery. It wasn't so easy to get the genes into the cells, and a lot of the efforts were focusing on these early tools of vectors to get the gene in the right place. And obviously there was a bit of collateral damage with these early technologies, and they had to be refined. But there was a bit of hype in those days, and the expectations were not really always realistic. Consequently, there was a lot of skepsis. And gene therapists as a community faced critical assessments by review panels, scientific peers, regulators, lay press, journal editors, and big pharma. And this created unprecedented adversity. And perhaps the only thing what one could do is to comfort oneself in some sort of philosophical wisdom, if you may. And luckily we could rely on some great philosophers to get us through these tough times, not the least the Dutch philosopher and freethinker Baruch Spinoza, a freethinker avant la lettre, who actually lived in Holland in the early ages of the Enlightenment. And I couldn't phrase it any better than Spinoza himself: be not astonished at new ideas, for it is well known to you that a thing does not therefore cease to be true because it is not accepted by many. And that pretty much captures the spirit of the field of gene therapy, let's say in the early 2000s. But things have changed, luckily. And there's been an explosion of gene therapy advances in preclinical studies, and certainly also some notable successes in clinical studies. This is a list of different diseases that have successfully been treated by gene therapy, leading to long-term therapeutic effects, perhaps even a bona fide cure. And the initial trials were focusing on rare immune deficiencies like ECTH1, but we see now that there is success also in neurodegenerative diseases, in congenital blindness, graft versus host, metabolic diseases like lipoprotein lipase deficiency, and even cancer. Cancer gene therapy is becoming very promising indeed. And last but not least, hemophilia. So where do we stand in hemophilia gene therapy? There's a few of important questions that we need to address here. Can we ultimately achieve a real cure of this disease by gene therapy for hemophilia A and B? Can we prevent the induction of neutralizing antibodies to the clotting factors? A major problem, as many of you know, following recombinant or plasma-derived clotting factor therapy. Are there any side effects that impact on the safety or the efficacy of gene therapy? And can this technology be improved further? Let me start off with the first question. Well, I'm definitely a believer, otherwise I wouldn't be standing here and I wouldn't have devoted 30 years of my career on this. But it's been indeed perhaps slow process, but as long as we know where we're heading, I hope we will eventually achieve that goal. So we focus on liver-directed gene therapy. Why the liver? Because the liver is the organ that normally, in which the clotting factors are normally expressed. So it was kind of an obvious choice. And Dr. Natwani and Dr. Davidov and colleagues, actually, and Dr. Tuddenham, among others, proved that it is possible to achieve long-term expression of factor IX in patients with severe hemophilia B. So what they did is they packaged a functional copy of the factor IX gene into an AAV8 vector, and then they administered, let's say, a moderate vector dose by peripheral vein administration to these severe hemophilia B patients. So the good news is that in many of these patients, they had sustained expression, which I believe now extends to more than 5 years, in the 1% to 5% range. So there's no doubt that this is a therapeutic level indeed. But there are some issues, however. First of all, there's still room for improvement. 1% to 5% is great, but it could still be improved in order to hopefully diminish the risk for bleeds even further. And there are some issues of liver toxicity which are not fully understood. And I just want to single out this patient here who has a surge in transaminases several weeks after vector administration, perhaps resulting from an immune reaction. And I will come back to that later. So the question that we were focusing on in our lab is how can we increase gene expression of factor IX or factor VIII so that we can actually lower the vector dose and perhaps get around some of these immune concerns that patients face when they are injected with these moderate vector doses. So we decided to go back to the drawing board and design promoters based on the premise that promoters that are highly expressed in the liver has some particular sequences in their promoter that confer high hepatocyte-specific expression. So we use complicated bioinformatics tools to actually sieve out these molecular denominators associated with high hepatocyte-specific expression. And these are clusters of transcription factor binding sites. So it's not so much the presence or the absence of such a factor, it's really how they all come together in a cluster that confers high expression. So we're trying to extract what nature has already done for us. What is the molecular denominator for high hepatocyte-specific expression? So we use those elements now to drive expression of factor VIII and factor IX, because actually the promoters, the native promoters for factor VIII and factor IX are not really that great. They're not really that efficient. And that was one of the reasons why we decided to adopt this strategy. Just to give you an idea and to demystify this a little bit, some of these elements are quite small. They're only about 70 bases long, and they bind quintessential hepatocyte-specific transcription factors like NHF1. So we predicted this based on the in silico genome-wide analysis, but we also confirmed this experimentally using chip assays. So the predicted factors do indeed bind on these elements. Do they work? Well, we took a factor 9 gene driven by two different liver-specific promoters, a weak one and a strong one. And then we cloned this hyperactive cis-regulatory module, as we call it, or CRM, in front of the promoter. So this serves as a molecular turbocharger, if you may, to boost the performance of the promoter. And you can see that we end up with a 100-fold or 10-fold increase in factor 9 expression in hemophilic mice that receive this vector. Now, I always make the analogy, if you start with a strong promoter, it's harder to make it perform faster. It's like trying to make a Ferrari go faster. But yet we were encouraged that we could, even in those cases, with a transthyretin promoter, which is quite a potent hepatocyte-specific promoter, still obtain fairly high levels of expression. There's no point having a potent promoter if eventually you lose specificity. If those genes are also expressed then in other cells, this raises possible safety and immunological concerns, particularly if these clotting factors were to be expressed in antigen-presenting cells. We and others had evidence that if that happens, you often break tolerance to the clotting factors and antibodies ensue. So we were really, let's say, pleased and reassured that the specificity of this promoter was not compromised. We injected a whole batch of vector, that's about 10 to the 12th. Normally we do 500 mice with that in a single animal, and yet we could only detect expression in the liver. So there's no leaky expression anywhere else, which we found reassuring. So we moved all the way from in silico to in macaco, and we treated two non-human primates with this vector expressing the wild-type factor 9. Actually, it was a codon-optimized version. And we could obtain 20 to 40% human factor 9 in these non-human primates. There was no evidence of any toxicity in these animals, which was also reassuring. So just to put things in perspectives, if you look back into the clinical trial where the same type of vector dose was used as in this animal, we actually managed to crank up the levels of expression at least 5 to 10-fold more than in the clinical trial. So from 5% to 40% max. But we think, based on the premise that we should use a vector dose that is as low as possible so that we can avoid any inadvertent immunological reactions, we decided to go one step further. And the paper was published by Paolo Simeone and colleagues, and Volder Arruda and many others in the New England paper that was very inspiring indeed, because they found a patient with a gain-of-function mutation in the factor 9 gene resulting in thrombophilia. And they coined this mutation as the factor 9 Padua, which obviously was identified in, you guessed it, Padua. So clearly that's opened up new perspectives, and it rang a bell. We decided, well, maybe we can use this Padua version, this thrombophilic factor 9 version, to treat hemophilia. And this is a busy slide, but just wanted to go through with it. These are just two different vector doses. And we injected hemophilic mice with either the wild-type factor 9, and you can see here factor 9 activity in black and the antigen levels in white. So there's a 1-to-1 ratio between activity and antigen if you use the wild-type protein, obviously. But if you use the Padua gain-of-function mutation, you have a lot more activity for the same amount of protein. That's what you see here, and we confirmed this in two different vector doses. In fact, when we quantitate this, it's about 5 to 10-fold more activity after doing gene therapy with this Padua version than with the wild-type version. We've actually taken this also into other vectors, not just AAV, and I will not go into the details about the vectorology. Suffice to say that this vector actually integrates in the genome, whereas the AAV vector does not. The AAV vector is a non-pathogenic is derived from a non-pathogenic virus, adeno-associated virus, but it largely doesn't integrate. So if you use an integrating vector like lenti, we obtain therapeutic levels, and then I say we actually refer primarily to the team of Luigi Naldini as well as Tim Nichols, who actually took our preclinical data in mice all the way forward to the dog model. So in a nutshell, what have we achieved? We know that it's possible to improve the cassette by this computational approach to identify new elements that that increase the performance of the promoter 5 to 10-fold, but we can also use this Padua gain-of-function mutation, which also increases the activity 5 to 10-fold, and we've put it all together into one vector. Now, these elements are quite small, these regulatory elements. In fact, there was a commentary on our paper, and I quote, "A David promoter with Goliath strength." And I have to say that our study definitely didn't reach any biblical proportion whatsoever, but I thought it was a nice quote to emphasize the fact that it's a small promoter that's nonetheless quite efficient. Normally, you compromise on activity if you use small promoters. Now, this Padua gain-of-function mutation has now been used in a number of clinical trials that I just wanted to summarize here, and it's reassuring to see that the work that we initially published in mice and dogs is now being confirmed also in human trials, and many of you in the audience actually contributed in one way or another to some of the results that I will talk about. I just wanted to start off with one of the first trials using the Padua factor 9 version, which was conducted by Baxalta, and they obtained peak levels more than 50%. Not all the patients responded in the same manner, but it was reassuring that actually in this study, some of the patients expressed the highest level of factor 9 activity than had ever been reported after gene therapy in a clinical trial. But this is not the only study. It's actually another study by Spark Therapeutics where they also use this factor 9 Padua, and they had sustained levels in the 30 to 40% range with diminished clotting factor usage and no sustained elevation in liver enzyme levels. So this proves once again that using this gain of function is very interesting indeed. Just to compare with the doses of vector that were used in the Natwani trial, they used less vector, 5 × 10^11, which is 4 times less, but they had 30% activity, which is 6 times more activity than in the initial trial. Unicure also used a new AAV-based vector, AAV5, but they didn't use the Padua version. They had about 5% of sustained factor 9 activity, also diminished factor 9 usage, and transient elevation in liver enzymes, warranting steroid treatment. So clearly the field is moving forward, either with Padua versions or with alternative serotypes, or both actually. I forgot to mention that in this trial, the Spark trial, a new capsid was used. What about factor 8? It should be easy now that we know that it can work for factor 9. Well, I wish it were true. With factor 8, everything seemed to be so much more challenging. Particularly if you use AAV vectors, they hardly accommodate large transients. And factor 8 is, of course, relatively large. The cDNA is 4.3 kb, and it barely fits in an AAV vector. So we're having some issues of packaging. Nevertheless, it is possible to package, and I will show you some of the results. But for this, we also needed to do some tricks, and we collaborated with John McVeigh and Natalie Ward and Adrian Driessche on a lentiviral concept where we use codon usage optimized factor 8, resulting in a 40-fold increase in expression. Now, this codon usage optimization doesn't alter the sequence of the protein. It just results in a more optimal usage of these codons, resulting in a higher level of factor 8 protein. We've actually plugged this into our AAV vector, again using our small turbocharger promoter that results then in sustained expression of factor VIII in the physiologic range using fairly low vector doses. Recently, BioMarine reported some exciting results at the World Federation of Hemophilia meeting in Florida, where they actually used a B-domain deleted factor VIII packaged into an AAV5 vector, and they zapped a relatively high vector dose in severe hemophilia A patients, and they obtained a mean factor VIII level of 146% at 20 weeks, which is fairly robust indeed. In fact, 2 patients, believe it or not, but had levels above 200%. The median annualized bleeding rate fell to 5 from 20, but there was a transient rise in liver enzymes, pointing to some level of liver inflammation or liver toxicity, warranting tapering doses of steroids. One patient actually had sustained an increasing ALT, so it'd be interesting to see how this eventually plays out. But clearly, this shows that also for factor VIII, it's possible to obtain very robust efficacies, although of course one has to try still to lower the vector dose in order to minimize any potential immune consequence vis-à-vis the vector. So what have we learned from all this? Well, if you look at mice, dogs, non-human primates, the expression is sustained when you use an AAV vector. Now, I told you earlier that these vectors do not integrate in the genome of the target cells, so the therapeutic gene just floats around in the nucleus. It doesn't integrate, mostly not. And yet we can obtain sustained expression for the entire lifespan of these preclinical models. The experience in patients is, of course, more limited, but going back to the Natwani trial, this expression is sustained for at least 5 years, and there was an earlier trial many years ago where the factor IX gene was delivered in the muscle, resulting in 10 years of expression. So will the expression be lifelong? Well, we don't know for sure, because obviously Homo sapiens is different from these species, but at least it is quite encouraging that we can obtain fairly prolonged expression. One issue that is very important for all of us is the issue of inhibitors. Will gene therapy increase the risk of developing inhibitors or decrease that risk? Well, again, we don't know the answer to that question yet. More studies are needed for that. But just wanted to share with you some of the work that we and others have done in mice, and this has also been confirmed to some extent in larger animal models, and that's the following. If you take a mouse and you inject this mouse with factor IX and adjuvant, these mice will develop antibodies to factor IX, no doubt. So this is a very provocative immunization, obviously, if you use adjuvants. Now, if we treat these hemophilic mice by gene therapy, regardless of the vector we use, AAV or lentiviral vector, regardless of the transgene, factor IX or factor IX Padua, even if we use factor VIII, we can achieve immune tolerance to the clotting factors. This possibly involves regulatory T cells, so we couldn't detect any antibodies against these clotting factors. Not always, there are some other factors that play a role, but in many cases it is like that. Even if we challenge these mice that had undergone gene therapy with recombinant factors and adjuvants, which should give rise to antibodies, well, still, they did not develop inhibitors, which is very reassuring indeed. It tells us something about expression of these clotting factors in the liver. Perhaps it predisposes, indeed, to the induction of regulatory T cells and immune tolerance. Are there any side effects that impact on efficacy or safety? Actually, I forgot to mention one thing, and that is also important, that if you express these clotting factors by lentiviral transduction in hematopoietic stem cells, the group of Bob Montgomery and colleagues have shown that even in the face of inhibitors, you can still achieve hemostatic correction. So in many ways, gene therapy offers new perspectives for treatment of patients with inhibitors. So are there any side effects that impact on efficacy and safety? Well, yes, and I wanted to come back to this patient in the earlier liver AAV trial. You can see here in yellow the factor IX levels, and in green the surge in ALT that was apparent in many of the patients, but certainly also in this one, requiring tapering doses of steroids. So what's going on here? Well, to be honest, we as a community don't fully understand, but it's possible that it's related to T-cell responses, whereby the vector enters the hepatocytes, and the vector components, shown in green here, the capsids, are presented as antigenic peptides on the surface of these hepatocytes so that they will then become a target for immune attack by T cells. So these CTLs, cytotoxic T cells, would then recognize these cells and clear them, resulting in transient expression. But if you subject these patients to transient immune suppression, you can mitigate this response such that despite the presentation of these peptides on the surface, you can still get sustained expression. So what's the morale of the story? Well, obviously, we need to optimize the vector so that it is safe but efficient. And like for any therapeutic, be it a gene therapy approach or a more conventional therapeutic approach, it's always a matter of finding the right balance. If you use a subtherapeutic dose, well, this is not going to be that useful, but it's going to be probably very safe. But if you increase the vector dose, you will eventually start to see efficacy, but one has to be careful that one doesn't provoke untoward side effects, which will eventually result in a negative outcome. And this is a complex picture because it's basically influenced by intrinsic factors, vector potency, transduction efficiency, and the vector dose, and extrinsic factors like inflammation, comorbid conditions, alcohol, age, etc. So all these factors may influence the outcome, and that's perhaps also why some patients respond better to the gene therapy than others. There's quite a bit of scatter in some of the trials. What we want is an efficient gene delivery system that gets you where you want to be. This car may not get you where you want to be. Believe me, I had something similar when I was a student. It didn't go that far. But we want to go here, but we certainly don't want to crash land because then we'll be back to square one. So as a community, we're really trying to sort out how far we can push the envelope. What would still be a safe vector dose that is efficacious and can give us long-term therapeutic effects. With the first trials, we're starting to get an appreciation of how far we can go with this. Can this technology be further improved? Well, I think so. I think we're all using AAV vectors that are based on AAV serotypes that are present in nature, AAV8, AAV9, AAV5. But we can also engineer these capsids so that we can enhance gene transfer and perhaps again lower the vector doses. We can increase the hepatocyte-specific gene expression, which is what we and others have done. And we really need to get a handle on these T-cell responses against the gene-modified cells. Now, these are not responses directed against the factor IX antigens. Let me be clear on that point. It's directed against the gene delivery vehicle itself. So we need to get a handle on this. And many of us are actually seropositive because of natural exposure to these AAV antigens. So if you're seropositive, any gene therapy attempt with AAV will fail. So can we render these AAV transduced cells invisible to immune attack? So can we solve the T-cell problem? And we decided to take a fresh look at this by developing what we coin as immune stealth AAV that can still deliver their therapeutic cargo to the cells, but whereby the gene-modified cells would not be recognized by the T cells. So does it work? Well, to some extent, what we did is we altered the capsid of these gene delivery vehicles in such a way that the antigenic peptides are then not presented on the MHC class I molecules of the transduced hepatocytes, in contrast to the conventional AAV vectors that everybody is using, where you can get this potential T-cell immune response. As you can see here, this is just an assay determining the T-cell reactivity vis-à-vis the transduced cells, comparing the stealth vector with the non-stealth conventional vector. And you can see that the T-cell response is drastically reduced when we use these stealth vectors, which, by the way, do not compromise in terms of efficacy. They're just as efficacious as the non-stealth counterparts. So perhaps instead of adapting the patient to the vector, we can adapt the vector to the patient, and we may not need immune suppression, or perhaps a much more milder dose. So all of this bears relevance to gene addition, conventional gene therapy, where the mutated gene of the patient remains unaltered. But could we also fix that broken gene? And this holds true not just for hemophilia, but for any genetic disease by gene editing. Now, this is easier said than done because obviously the genome is huge. It's 3 billion bases long, and you're talking about fixing one mutation in these 3 billion bases. It's like looking for a needle in a haystack. And that's challenging indeed. In fact, you may end up with some off-target effects. If you're trying to fix the gene that you want, you may also do other things that you don't want. Perhaps there's some hope with some emerging technologies that many of you may have heard of, which is the CRISPR. CRISPR-Cas is actually an emerging gene editing technology that allows us to site-specifically modify the genome in a very specific manner. There are other technologies out there, zinc finger nucleases, etc., but these CRISPRs are really relatively easy to design, and as a concept, it's very attractive. It was actually used in nature because bacteria defend themselves against invading pathogens by cutting the DNA of the invading bacteriophage in a very specific manner. And this work was actually, some of the validation studies were done by Charpentier, Doudna, and Zheng, and actually paved the way to a lot of fundamental studies, but perhaps also to new types of clinical trials. Now, how does it work? Well, I don't have time to go into great detail. Suffice to say that you need a protein, Cas9, and a guide RNA, which is a small RNA that brings this protein, which is an endonuclease, exactly where you want it to be. And it's going to cut at that very location, and the location is determined by the specificity of the guide RNA. So depending on how you change the guide RNA, you will bring that Cas9 to a different region in your genome. And what you can do with that is really quite amazing. It's a versatile tool. You can induce double-strand breaks in your DNA, which are then repaired by non-homologous end joining, or you can induce double-strand breaks in your DNA, which then improve the efficiency of homologous recombination a million-fold. So you can really start to think about gene repair. So let me just share with you some of the recent data that we've obtained in the lab as a proof of concept. We actually wanted to address a very basic question. Can we go after the factor IX gene? Can we target the factor IX gene by this type of somatic gene editing? And when we started the work, it wasn't really clear. There was no publication that you could actually do this. And now with the work of Zhang, it's starting to become apparent that you can. So what we did as a proof of concept is we actually tried to make a wild-type mouse hemophilic by inactivating the factor IX gene. So that was the first step. So we delivered 2 vectors in these mice, one vector encoding the Cas9 nuclease, and another vector which is specifically designed to express the guide RNA that binds on the factor IX target sequence. So we injected these vectors in these mice in the hope that we could inactivate the factor IX gene, and if that happens, then our wild-type mouse should become a hemophilic one. Of course, if you think of applications, you want to do exactly the opposite. You want to turn a hemophilic individual into a wild type. So the repair is more challenging, but we just managed to achieve the first step. So this is promising, but it's not quite ready yet for prime time. We need a lot of vector. A high vector dose is required, and transient expression of some of these components would be preferred. But given these caveats, I still wanted to make the point that even though it's challenging to find a needle in a haystack, it is possible to find that needle. And that needle, in our case, is the factor IX gene. And this is the factor IX activity in our mice, so wild-type mice, taken at several time points, about 100%. And when we do the gene editing, the somatic gene editing in the liver, you could see that the levels of factor IX drop significantly. So it's not completely zero, and that points to perhaps some of the limitations of this technology, but still it's quite significant indeed. We had a 60% drop in factor IX activity, and of course these mice did bleed more frequently than the non-treated, than the non-injected counterparts. So this tells us that it's possible to actually go in vivo and target the gene of interest. This may be interesting to some of you working on other genes, on complex phenotypes, where you can actually inactivate that gene without having to go all the way to generate a new transgenic mouse model, de novo, which takes a lot of time. Now, is it specific? What about these off-target effects? Well, we try to quantify this using complicated genome-wide techniques, and we had like 60% targeting in the desired exon, exon 1 of factor IX. It depended on the type of exon that we were targeting, depending on the type of guide RNA, so there is indeed some optimization warranted. But we didn't see any off-target effects anywhere else in the genome when we use computational programs to predict where else in the genome these protein may cut. So at least it's quite specific. And now, of course, we're trying to repair genes and see how this goes. Now, I think, I'm not saying that gene editing is the way to go for hemophilia. I think certainly this isn't ready for prime time, but it's an interesting way to try to approach the problem, and perhaps it may be useful in some applications and not in others. There was a paper published by Dr. Kim from South Korea where he actually used it to correct the inversion in factor VIII, which I thought was quite elegant indeed. So the morale of the story and the journey that we have embarked on many years ago is that we can try to improve the technology of gene therapy at different levels. We can improve the vector, we can improve the gene by codon usage optimization, by gain-of-function mutations, or by combinations of the above, or we can improve the promoter. And when all comes together in the most optimal configuration, you can then conceive clinical trials with low vector doses, and that will hopefully be also safe enough in patients. And I'm definitely very encouraged by the way the field is going. In many of the clinical trials, there's long-term therapeutic effects, and we can learn from what we observe in these trials, going back to the bench, improve the tools, and going back into patients. So last but not least, I'd like to thank the folks who've contributed to this work. This work was done primarily in Brussels, in the Department of Gene Therapy and Regenerative Medicine, but also in Leuven, in the Department of Cardiovascular Sciences, where I have another appointment. And actually, I'd like to single out Marini Chua, who's been with me on this journey since the beginning. She's still bearing with me. I don't know how she does that, but still, hopefully we can continue to work together. And I'd like to single out Nisha Nair, who's been extremely helpful as part of her PhD in this work, as well as many other fantastic individuals and our collaborators in this work. Last but not least, welcome to Brussels. Some people think it's a hellhole. I respectfully disagree. Come and visit us, and we have a drink on Grand Place in Brussels. Thank you very much.
Thank you very much, Professor vanden Driessche, for this excellent lecture, very informative, and thank you for sharing with us this very new data. So we have a couple of minutes for discussion, and please do not hesitate to make comments or questions.
Menno Huisman from Leiden, and I enjoyed your talk greatly. We talk a lot about personalized medicine, and you hinted on this because you said there were external factors, including prior hepatitis infections. But is there another way that you can predict the effect of gene therapy, and is that maybe a way to go? In otherwise, is the gene buildup of a patient predictive of how that patient would react on either the two ways of gene therapy?
Can you just repeat your question a little bit? I was a bit affected by the acoustics.
I was interested whether you could predict in a patient how that patient would react on one of your ways to put gene therapy, and not only because of nurture, but also of nature of that very patient.
Well, to some extent, we know that there are certain criteria that have to be met. If some of the patients have a low titer, even a low titer of pre-existing antibodies to the vector that we use to deliver the transgene, then it's not going to work. And it may seem obvious at first glance, but actually it isn't, because many of us assumed that the neutralization of the vector would only occur at much higher titers. This turned out not to be the case. In the recent trial of Baxalta, now Shire, there was quite a bit of variation from patient to patient, and it's not really fully understood why some patients did react fairly well and gave sustained expression, and why some of them didn't. They looked at some of the immune reactions, the T-cell immune reactions, and it didn't fit 100%. So, whereas in some trials it seemed to fit, in other trials it don't. So, to make a long story short, we don't really know exactly all the confounding variables that would predict the outcome in a given patient. We know of some, but there are others that we simply don't have a handle on. Perhaps we do indeed have to do more exhaustive characterizations of the immune system, or GWAS studies may also be helpful in this respect.
Thank you. Any other questions from the audience? Maybe I can ask you something that is interesting from clinician point of view. 30 to 40% of patients have naturally occurring antibodies to AAV vectors, so we cannot treat them with gene therapy. Do you, as molecular biologists, have any response to that, any kind of research, how to refine vector and to make it invisible to antibodies?
So the point is, what happens to those patients who have pre-existing antibodies to the vector following natural exposure to the cognate virus? There's two possible ways of approaching this problem. One can change the code of the vector. You can use another serotype, which is less cross-reactive to the naturally occurring ones. And I believe that AAV5 is a bit more favored in that respect. I also know that from the trial of Spark Therapeutics that they use a variant in the capsid that is less likely to be neutralized by these pre-existing anti-AAV neutralizing antibodies, which are indeed quite prevalent in the human population. So you can change the capsids either by engineering or using other naturally occurring serotypes. Or you could also use another vector altogether, and that is the lentiviral vector. The lentiviral vector doesn't face the same type of problems like the AAV vector. In fact, as I have shown in the dog model, you can get sustained expression of clotting factors with lentiviral vectors following hepatic gene delivery. Since patients are not exposed to wild-type HIV, unless, of course, they're an AIDS patient, you don't have to be concerned about the issue of pre-existing immunity if you use lentiviral vectors, as much as you would have to be if you use AAV. So sometimes a radical approach and changing vector altogether is one way of going about. There's a lot of other tricks we can do, but this will bring us too far. This is still a work in progress. It's one of the challenges in the fields.
And just one more question. What about risk of insertional oncogenesis with using of lentivirus vectors?
I didn't understand your question.
Risk of oncogenesis when you use lentiviral vectors. It is higher. Is it higher?
Well, that's a very interesting question. So what is the risk of oncogenesis? If you use an AAV vector, 99.99% of the genomes of AAV do not integrate in the target cell genome. So as long as they don't integrate, you have no risk of insertional oncogenesis. But it's not 100%, so a very small fraction of those genomes do integrate randomly in the genome. And theoretically, one could think that perhaps if those genes happen to integrate in an oncogene, you may actually predispose that cell towards malignant transformation. So this is a theoretical consideration which we cannot 100% discount, obviously. And tumors has been seen in very, very sensitive tumor models in mice, but you really have to push the envelope. You have to use high vector doses, you have to use tumor-prone mouse models to actually see it. We certainly have never seen it in our mice with high vector doses, as did nobody else using the vectors that are used clinically now.
Thank you very much. Are there any more questions? If no, thank you very much for your lecture. There is a sign of appreciation from organizing committee.
Thank you. Thank you very much. Thank you.
Thank you all for participating, and I would like to invite you to stay with us for a few minutes to close the Congress officially.